And they look a little bit like this. The full paper is in review right now. But these are phylogenetic trees, not phylogenetic trees, but these are trees that sort of align by principal component analysis the different patients. And the most important thing to see is that healthy, lupus, chronic fatigue, they're all sort of lining up all over the place. They're not clustering one side of the diagram or the other. And that means that what we're finding in terms of sequences in these specimens are not differentially distributed between these groups. And on the bacterial side, if you take a look at what you actually pick up, because these are highly sensitive things, these are mostly water-loving things that are known to occasionally sit in the reagents, and they were equally distributed within our clean controls and things like that. So what you get with metagenomics is you get an awful lot of stuff, and it's very important to be clear about what's differentially distributed. Well-controlled experiments are important because life forms are everywhere. They're even in the reagents that you use in the lab. Similar things with viruses. This slide didn't come off too well, but again, nothing really exciting going on there. So what we're reporting in the paper is that we're not finding big differences in terms of the bacterial or viral content of blood. Yeah, but this isn't the end of the question when it comes down to infectious diseases and ME, because we've not interrogated every tissue in the body that might be relevant. So it's really important to say that. Now on the gene expression side, Jérôme Bouquet, a nice Frenchman, he's now down in UCSF, he's gone through things for us, and what we are finding is that the RNA quality is really good in the specimens. We know that we can find gene expression differences because we're finding them in droves with lupus. And so the SLE group has been a wonderful positive control. On the other hand, there's relatively few differentially expressed genes. This is in a cross-sectional study, healthy state, between people with ME/CFS, the alternately defined Lyme group, and healthy. This is just gene expression. It's not the totality of metabolism or anything else along the way. Now, Charles and Jerome had published a paper that had found 2,000 differentially expressed genes in Lyme disease, not one of which is differentially expressed in the alternately defined Lyme or over to the people with ME/CFS. So it's an important negative in terms of the transcriptomic side of the equation. And we've been working hard to sort of make sure that we're not just looking at the lab stuff in isolation. We're trying to tie it together with the clinical parameters, the epidemiological findings, the other covariates. And so we're currently doing some sub-analyses to see if there's anything else we can learn by correlating the transcriptomics with fatigue severity and other variables along the way. Now, the exercise testing in ME/CFS, you've probably followed the story with interest, and this is why we got together with the group in California. What we're interested in doing as much as possible is to use it to objectively measure physiological markers. Now, you'll remember that Stevens and other groups have made observations that on the second day of 2 days of exercise testing, there seems to be a decrement in anaerobic threshold in some people with ME/CFS, an objectively measured decrement. Now, this hasn't been replicated in every other study, but it does allow the possibility of perhaps identifying discrete physiological subtypes of ME/CFS, so that if you're doing other studies, you're able to sort of subtype them or stratify them based on their response on cardiopulmonary exercise testing. But to me, we were also interested in it's all well and good seeing people when they're in a relatively good state of health coming in, but we haven't been visiting people who are sick at home. We haven't built that into our studies yet. Some of you are doing that, and I commend you for doing that. But we also have a lot of people saying, look, I'm up and I'm down. I'm quite prepared to have you take a look at me when I'm down. And so we can actually have a standardized measurable experiment, if you will, that will allow people to go through some exercise and maybe experience symptom flare afterward. So that's what we've been doing. And the study design, and this is the one where Merrick really has helped us out, because the original funding we got on pilot funding from the NIH allowed us to run the exercise testing and to collect some specimens before and after, but didn't fund us for enough follow-up to go out a week and longer in terms of visiting people at home, collecting their specimens at that time, and seeing what gene expression is, much further out through to recovery. So basically what we're doing is people are coming in, they're going through the same sort of screening that we did in our other studies, all kinds of things to exclude other diagnoses and things. They're coming in and they're doing this bicycle ergometry test on the first day, and we're collecting their blood beforehand. We're doing it on the second day so that we can see if any of the observations that Stevens and others had can be replicated in our population. But then we're following people out during the time when they are having symptom flare. And we're doing this in ME/CFS, and we're doing it in healthy but deconditioned controls. So people who are not very active at all. And we put Fitbit sort of testers on people so we can actually measure the steps along the way. So, so that study is at the point where Jerome just sent me the data. So I'm actually analyzing it on this trip. And we hope to be able to get something together very soon to see whether we've got any pattern of gene expression there that's different. And we've got the exercise testing data down to Stacy, and we'll see whether we've got people who seem to be subsetting as well. So I wish I could tell you the complete story because Merrick, but we're still analyzing it. Now, I wanted to tell you a bit about immunosignature work. Essentially, you've had these high throughput technologies in terms of genomics, where revolutionized approaches to gene sequencing. But when it comes down to defining what's going on with antibodies, it's been very hard to take a look at the totality of antibody expression. So Stephen Johnston at ASU has developed these microarrays, peptide-based microarrays that will have on one version, 125,000 peptides, on another 330,000 peptides on a chip. Now these are randomly selected from potential antigen space there, but it's enough to give you a very good broad overview of where antibodies are being expressed. And so these chips have been used to make early diagnoses of valley fever in ASU. They've been used to interrogate vaccine responses, and they're being used in cancer research to interrogate immunosignatures associated with specific malignancies. So they're fairly mainstreamed, and all we did was get together with Stefan to say, could we begin to look at this in chronic fatigue? So we'll be reporting next month and putting the actual data up in Fort Lauderdale that we are seeing some differences between ME and controls in cross-sectional study, and it's enough that it's helping to distinguish a sizable subset of the folks with ME from controls. So we think there may be some value in this in terms of the potential for a disease signature. But we've been working with Fluge and Mella as well, because as you know, some of their people responded to B-cell depletion via rituximab in their studies, and some of them didn't. And the clearer differences in immunosignature that we think we're seeing in our initial work are between the responders and non-responders, which makes a bit of sense if the responders have a specific pathophysiology along the way. But yeah, I think this is an interesting technology. We'll tell you more about it next month. But we consider this as hypothesis generating. So to prove these sorts of things in the longer run, we're trying to find some funding with Fluge and Mella to test their entire phase 3 group and make a prediction about response and test that prediction along the way. So that will prove whether this is of clinical value. And we also think that this is something that needs to be referred to the prospective cohort studies. So what I wanted to leave you with was our thinking about where causal inference really needs to go next. We've had a lot of wonderful cross-sectional studies that are beginning through the omic applications to show things. And I wanted to remind you that we have these cohort studies, and the UK is as well off as any other country on the planet because you have wonderful cohort studies and probably the best biobanking of any nation state on the planet connected with those cohort studies. But we've also got a cohort study put together for the purposes of cancer in Canada that's 300,000 people strong, and it's coming up for its first follow-up. And so I spoke with John Spinelli, one of the local PIs, and we've got questions in there about new onset ME. So after 3 years of following up 300,000 people, there will be people with a new diagnosis. And of course, those people have biobanked specimens when they were healthy. So you start in a nested case control study with a cohort population, you follow them up for some years, some develop disease, and it doesn't have to be ME, it could be cancer, it could be anything else. And these are the cases in your study. The rest remain disease-free, and you can't necessarily study them all, that's very expensive, but you can do your multiple omics on a representative subset of control. And this way you can not just make a comparison between the cases of controls, but you can do the before-after in each way. So that allows us to say, well, is this metabolomic profile of Navios a result of forced inactivity because of something else driving the disease state, or is it causal, or potentially causal? And that's why we need these studies, and we need to marshal these cohort studies along the way. We should focus on platforms showing current promise. So the current cross-sectional studies are really useful. So the cytokine networks, the metabolomics, the immunosignature. I love the feces. I think it's going to be really important along the way. So back to the winning horses where we're seeing some differences going into those studies. And is this easier than a moonshot? It seems like a lot. Well, the cohort studies are already up and running for different purposes. The omics work is already happening. And I feel it's just a matter of joining these efforts together. And with that, I wanted to mostly acknowledge everybody who's participated in our studies. It's been a lot for them to come out and do this, and our funders, and particularly ME Research UK for helping to bring this along. Thank you.
Thank you very much, David. So there's time for questions.
Yes.
So.
David, yes. Hi, Zahir Nali from the Southern BCFS Initiative, the great talk, David. Do you care to comment a little bit on the cytokine studies that you presented, no difference you saw compared to the Hornig, Lipkin? Were these also divided in early versus late stage? Have you done that specification as well?
Yeah, it's a perfect question. So we did take a look at that, and actually a student back reanalyzing by duration of infection and finding no difference. But the reason that we could not find something that Hornig and Lipkin could is sample size, which is why these efforts of yours to have bigger groups is important. Hornig and Lipkin had enough people in the first 3 years. We only had 3 or 4 in the first 3. You can't make any inference about that at all. So if you're saying that cytokine networks are going to be different in the first 3 years, nothing that we have shown contradicts that. We just didn't have the ability to interrogate that for the first 3 years. But as I mentioned, I thought that the work they published on cerebrospinal fluid was even more important because I think it's very important for us to understand what's signaling what within the central nervous system.
I comment there too. I think some of the differences that the Lipkin study reports are extremely small. I mean, they are statistical differences. What are their biological significance is something else.
I can't remember if I learned this from Paul Cheney or Byron Hyde, but regarding your hand grip exercise, a good diagnostic test for ME is to do this.
Yeah.
Okay, and this. So the thing is that I'm exercising my grips, but also my heart is working.
Yeah.
So the question is, when you consider the perceived effort and the measured effort, are you just working out the energy in the muscles of the arm, or do you take into account the fact that the whole body is an ecosystem and is working on this?
I think that question is great, because what the submaximal thing does is it's really measuring what's going on in that particular muscle group and what that muscle group is doing there, and also the oxygen that's offloading in that muscle group. The value of maximal exercise testing, where you get on a bicycle ergometer or a treadmill and you're measuring the gases and everything else, is that you're able to measure heart rate, blood pressure, gas exchange, and so forth. You're able to objectively measure the physiological outputs, which are really ultimately the outputs of the mitochondria along with. So I'm a firm believer that if we're messing with exercise testing, at the moment there's no substitute for maximal exercise testing, to anaerobic threshold if possible.
Anybody else? So let me ask Dave. You commented on this hit and run hypothesis and looking for an infectious agent. So does the infectious agent still have to be there? I mean, there are good examples in neuroscience, my own world, where infection in utero, there's a very strong view that at least a significant number of mental health diseases are caused by in utero infection, but there's no infectious agent left when the disease develops 20 years later. So is this the right place to look?
The hit and run hypothesis, that's always been the problem with it. I mean, the very hypothesis states that you get exposed to a trigger. It may not be with you any longer. I mean, right now, for example, take a look at that group of people with getting Guillain-Barré syndrome following Zika virus infection. The virus is no longer with them necessarily at the time that they have the autoimmune attack on their myelin. It's gone. So how do we know they've had Zika? We know they've had Zika because we can get a serological response to the virus, which is going to remain with them for a very long time. So back to exploring the hit and run hypothesis, it's been very much one of saying, okay, are we playing 20 questions? How can we do serology for Epstein-Barr virus, for hepatitis B, for chlamydia pneumonia? You could go on, and you could run out of time and money doing ELISAs. So the idea was, is there a maximum, a way that you can look for an immunosignature more globally in the same way that we do high throughput sequencing with genomics? And that's what I think Johnston and the Phoenix group are onto. They're onto the beginnings of a technology that allow us to do that. And so what we can do is we can look at a unique signature for a given disease. We can look at the peptides that comprise that signature, and we can try and relate those back to specific antigens. Now, going back to specific antigens on this antibody technology is much harder than going back from a gene sequence to a specific gene, because there are so many different permutations and combinations of amino acids that comprise parts of protein. But it's the beginning. And so it would allow us to talk about antibodies that are directed against pathogens, but also directed against self, against proteins that are part of you. So if you go with Flusion-Mella and the autoimmune hypothesis, it's important to look that way as well.
It's a powerful way in.
Yeah, just a quick question. So, Carl Gordon from Oxford. Can I ask you about your RNA-seq data on the post-exercise?
Yeah.
So what cells were you actually looking at? So they're not muscle cells, they're blood cells?
It's blood. Yeah.
So could you look at the muscle? Is there a way of looking at the changes in the muscle in terms of the RNA sequence?
I need to speak to somebody who regularly does muscle biopsies to see how minimally invasive one could be on that. The challenge with looking at other tissues has always been collecting specimens from other tissues. But it's also interesting that there is spillover into the blood from compartments, and so it's important for us to try and understand that as best we can. At the beginnings of applying a technology in an area like this, we really have to go with the specimens of convenience and try and gain the experience and then move a little deeper as we go.